Okay, welcome back to the Classics of Immunology Journal Club. Before we get started, don't forget to like the video if you'd like it, and click on the bell to get notified for more. And also scroll down on the YouTube site and check out my website. Particularly look at the writing tab because that's where all the classic papers that I've picked out are there, and you can just sort of read to your heart's content there. Today, we're in the 80s now, in the 1980s, and we are really heavy into how T cells work, how they do what they do. And of course, in the 60s and the 70s, experiments were done in cellular immunology to figure out T cells did all kinds of things that they didn't know about before. You have to realize that it wasn't until the 60s and the 70s that T cells became a thing. Before that, they were not a thing. They were never discovered. And so as time went on, they found that they had antigen receptors just like B cells, and the B cell antigen receptor was surface antibody, and that was sort of nice because the immunologists had spent a half a century trying to find out the structure and function of antibodies. And so that was already done. With regard to T cells, we didn't know. And the other thing we found people found was that T cells were giving instructions to other cells. In particular, they were giving instructions to B cells to tell them to make antibody and also to tell them what kinds of antibodies to make and so forth. And in addition to that, the T cells would differentiate and become killer cells, and they developed assays for the killing mechanism and so forth. But exactly what was going on and how T cells did that, we didn't know. But the critical experiments that I think, and I attribute the whole thing to, was there's a series of experiments that were done in the 80s where for the first time we had monoclonal T cells. And I reviewed that paper. That was one of our papers back from 1979. That was key because the whole T cell compartment, populations of cells, were so complex and so complicated that it was very difficult, if not impossible, to study a function and relate that back to the molecules that the T cells were using. And so today, we're going to delve into for the first time in how T cells might help B cells make antibodies. And the paper that I want to focus on was in Nature from 1985, in May of 1985, by Junichi Ohara and William Paul from the NIH. And the title is The Production of a Monoclonal Antibody and Molecular Characterization of B cell Stimulatory Factor 1. And the background on this paper is that from about 1979-80 on, there were several different groups in the world looking at trying to understand how T cells helped B cells. And what they needed to have was a biological assay for the B cells making antibody. And so different assays were developed by different groups. And consequently, when they looked at the T cell for soluble molecules or factors that might help the B cells, they named the factors based on their own assay. And there were different kinds of assays. One was just proliferation of B cells. And so they talked about B cell growth factor. And then they had the kinds of antibodies that B cells made. They found that T cells were making factors that would cause the B cells to switch the isotypes that the B cells were making and favor one of the IgG molecules, IgG1, out of the 3 or 4 different kinds, and also IgE. Turns out IgE is important for allergy because that's the antibody that gives you an allergic response because it's triggering mast cells to release all of their products. So that was they called it a class 1 switch factor. And that was one of the B cell stimulatory molecules. Another thing that they found that T cells could do is they helped B cells express on their cell surface MHC molecules. And of course, the MHC molecules, there were two different kinds, class 1 and class 2. And they found that the B cells would activate a whole increased density of class 2. Now, that was important because the class 2 molecules on the cell surface became antigen-presenting molecules for T cells, and in particular, the helper T cells, the so-called the CD4 positive helper T cell category. You needed to have an MHC class 2 molecule plus whatever the antigen was to activate the T cell. So there's all kinds of things going on, back and forth, crosstalks. And of course, it was heady times and very competitive times. And people, they wanted to be the first to do this, that, and the other thing. And of course, competition breeds sometimes, nasty things happen. But we won't talk about that too much. Turns out that Junji Ohara was a postdoc from Japan who came to the NIH to be in Bill Paul's lab. And Bill Paul, just to give you a little background on him, he was a postdoc with Baruch Ben-Esraf when Baruch Ben-Esraf was at NYU, and Bill Paul had gone to medical school at NYU, and then he ended up working in Ben-Esraf's lab way back in the earlier days, in the late 50s, early 60s. When Ben-Esraf got to be more and more established and famous, he was offered a job to go to Harvard to become the chairman of the pathology department up there, and also at the Dana-Farber Cancer Institute in Boston. And soon after, he'd gone from NYU to the NIH and only been at the NIH a year or two. Then he was offered the job at Harvard. And so Ben-Esraf left, and he had already taken Bill Paul there from New York to Bethesda to be at the NIH. Well, it turns out Ben-Esraf recommended Bill Paul to take his place there at the NIH. And so he became the head of a laboratory, which is sort of like being the head of a department in a university. So it's a lot of responsibility for a young guy, and he was just a young guy. But he's then stayed there his entire career. He was very smart because that's where the money was. And so, when in the 2008 financial crisis, when Wall Street and the banks went belly up and had all kinds of problems, I said to myself, well, that means that the new Wall Street is going to be Bethesda, and in the NIH and the government, because that's where the more and more money's going to be there than any place else. And so Bill stayed at the NIH, and he had been there, I don't know, at least 10 years, maybe longer when they wrote this paper. And one of the things about this paper is it's about a monoclonal antibody to a B cell, to a T cell derived factor that helps B cells make antibody. And I had, last week, we reviewed one of my papers on making a monoclonal antibody to interleukin-2. And when we did that, was looking for a place to get it published. And one of the places I sent it, sort of before I submitted it for publication, was I sent it to Bill Paul because he was a member of the National Academy of Sciences. And I wondered whether or not we could publish it in the proceedings of the National Academy of Sciences. And so he read the whole thing through, and he wrote back saying, well, it's a great paper, but it's really too long. They have a page restriction in PNAS of only 3 or 4 pages, essentially, which I knew. Anyway, so we didn't publish it there, but he knew all the things that I told you last week about how to make monoclonal antibodies to cytokines. And this is what this paper is. He did exactly the same thing that we had done, but this is now 2 years later. Our paper came out in 1983. This is now 1985. In the introduction of this paper, they talk about what is B cell stimulatory factor number 1. And they said it was originally B cell growth factor, which was a T cell factor that stimulated B cell proliferation that was activated via an antibody to the surface immunoglobulin of B cells, the mu chain of the heavy chain of IgM. But what you had to do is to tighter down the concentration of the anti-mu so that it was suboptimal. And then you would add on your factor, and then you would get robust proliferation. And so that was the assay for this. That was his assay, or their assay, for B cell stimulatory factor or B cell growth factor. But as I mentioned, this same factor, what it looked like, the biochemistry was going on and they were trying to sort all these things out. It looked like this same factor was responsible for activating B cells to express a large density of, a high density of MHC class 2 molecules, and also looked like it might be responsible for the switch in immunoglobulin production to predominantly IgG1 and IgE. So it looked like it might have been the switch factor as well as the class 2 stimulator and the B cell growth factor. And this is just a wonderful molecule, according to the B cell people. So in the materials and methods, in order to get an antibody, a monoclonal antibody to this factor, they needed to purify enough growth factor, enough of the factor, to immunize the mice. And so to do that, because they were at the NIH, they had worked out, or other people had reported, that a particular mouse thymoma cell line that was called EL4, if it was stimulated with 4-ball myristic acetate, it would make this factor. So they basically just got one of their contract labs at Litton Bionetics, which was a defense contractor, really. And they would just make liter after liter after liter of conditioned medium containing this factor and then send it over to Bill Paul's lab. I was a little jealous of that because we had to make our own factor up at Dartmouth. We were just a little operation by comparison to this behemoth down there in Bethesda. So then they worked out a way that would, in 2 steps, they could get things. They could reduce the complexity of the proteins in that conditioned medium and also concentrate and semi-purify the B cell stimulating factor so that they could use that to immunize mice. And the 2 steps that they had some activated glass beads that when they mixed it with the conditioned medium, the factor would attach to the beads, and then they could then elute them from the beads in a concentrated fashion. And then they could pass that through just one purification step through HPLC, high pressure liquid chromatography, and come out with a highly concentrated preparation. It wasn't pure, but that was, they were banking on the idea that if most of the protein in this was their factor, that they could then use that to immunize mice. And so in order to immunize the mice, they used a protocol that was very similar to what we'd use to immunize mice to make antibodies to interleukin-2. Their first immunization, they didn't do mice. They were working in mouse factor. So they immunized rats, and they immunized rats with a certain amount of their semi-purified factor, 1.6 times 10 to the 5th units. They didn't know what a unit was there at that time. And they mixed that with complete Freund's adjuvant. They followed that and boosted these rats twice with the same amount of semi-purified factor in incomplete Freund's adjuvant, and then followed that up with a dose in saline, at which time, after 3 or 4 days, they killed the rats, took the spleen, and did the fusion with the same cell line that we had used, NS1. They then screened these. They then cloned out the hybridoma cells that resulted from this fusion of spleen cells with the myeloma cells, and grew up the cells first in 96 well plates, then in 24 well plates. And they devised an assay where they looked for rat antibodies that would bind to their factor and reduce the factor activity in the supernatant, which then they used their bioassay in order to detect their B cell stimulatory factor. We should go to the results, and I should show you on screen sharing on figure 1. And so this is the figure 1 has 2 parts, A and B. And so this is where they used, they've now used it and they've generated 2 different monoclonal antibodies. that they thought was to their B cell stimulatory factor. This is the assay in thymidine incorporations into the stimulated B cells using anti-mu plus the factor, and this is their positive control. This is a negative control for the rat antibody. This was a rat antibody to the interleukin-2 receptor, and it did not inhibit the activity. Whereas if they used their purified antibody, and as they increased the concentration of the antibody, they decreased the B cell proliferation in their assay here. Then this is another assay they did in order to look at the activity of their antibody. And of course, so this is the anti-mu plus BSF1 stimulating the proliferation of the B cells. And this is the same control that was used here and does not inhibit, so that they didn't think that they had non-specific inhibition. And then this is the two different antibodies that were decreasing the proliferation of the B cell compartment. Then they decided that they wanted to characterize biochemically their ability of their antibodies to recognize the BSF. And this is an SDS page of their so-called, or their partially purified HPLC purified preparation. You can see there's multiple bands here. They were keying in on two different bands. And here is a blot of using radiolabeled antibody of one of their anti-BSF1 antibodies onto this kind of an SDS page, transferred to nitrocellulose paper, and then blotted with I-125 labeled anti-BSF. And you can see that there's two bands, one at 20 and one at 14K. And then down here is a two-dimensional SDS page looking at the molecular weight in one way, and then the isoelectric point in the other. There was one spot on a 2D gel from the 20 KD band, and they couldn't find any spot that corresponded to the 14 KD band. That's what they said. Then in figure 3 is the same kind of a protein blot using the I-125 labeled anti-BSF here. And then this is doing an SDS page and fractionating the gel, and then looking at the biological activity. And it really all comes out primarily in one fraction, one band from the SDS page. Now they didn't take it any further than that. They didn't do what we had shown in our anti-IL-2 paper, in that they were unable to show that they had only one protein. They still needed to generate large amounts of their antibody, and they were able to show that it didn't block responses to interleukin-1, interleukin-2, or interleukin-3, and in other experiments, interferon gamma. So they figured that they had a pretty pure preparation, and they had an antibody that would react to it. So that's the end of the story here. And so back to the discussion of this paper. They decided that on the basis of the data that they generated in this paper, that they were dealing with a T-cell derived factor that was not IL-1, IL-2, IL-3, or interferon gamma. And so they were probably dealing with a unique cytokine. And they said, now, since we've got this antibody, or at least they had two antibodies, now we can use these antibodies to characterize both the biochemical as well as the biological aspects of this factor. What happened, and so that was it in this particular paper. And the reason I put it in the classics is that it was really one of the very first papers that defined at a molecular level one of the factors that helped the T cells made that helped B cells. Ultimately, the very next year, in 1986, two papers came out, and this is what happened subsequently after this paper was published. two different groups came out and they had cloned the complementary DNA for this factor and ultimately named this factor interleukin 4 because interleukin 3 had already been named. And that's another story that I won't go into today. But one of the groups had taken a lot of the lessons that we had learned from interleukin 2. They devised a very good assay for this factor. And this group was headed by Eva Sørensen from Stockholm at the Karolinska. And she got together with Tezuka Honjo in Japan. And actually, Eva went over to Japan and spent several months in Honjo's laboratory. Eva took one of her postdocs with her. And Eva and her postdoc did the biological assays for this factor. And Honjo's group did the molecular biology, and they ended up cloning the gene for what ultimately was called interleukin 4. There was another group at DNAX, which was a new biotech company that was started up Stanford and Palo Alto. And Kenichi Arai was one of the professors that moved over from Stanford. He was a biochemist to become the head of that institute. And Frank Lee was the primary person. And they also simultaneously or very, at the same time as Severance and Honjo cloned it. And so that was good. From what happened then subsequently is that a lot of experiments were done. In particular, Bill Paul's lab did a lot of experiments with their antibody in the course of 1985 and 1986, trying to sort out exactly what this molecule was doing. And so it turns out that interleukin 4 is also a T cell growth factor. It's not as a potent T cell growth factor as is interleukin 2, but it does cause T cells to proliferate. And it also causes T cells to make more of their B cell stimulating factor, the interleukin 4, which then causes the B cells to switch to immunoglobulin IgG1 as well as IgE. And the IgE is really important. Ultimately, as time went on, the receptor for all the interleukins were found out about and antibodies to them were generated. And now in the clinic today, the anti-IL-4A alpha chain receptor antibodies are very good in the treatment of intractable allergic problems like asthma and atopic dermatitis and other allergic diseases. And so all of this basic science that happened in the 80s has finally come to fruition as time has gone on in the 90s and the aughts and now in the 10s and 20s. So I'll stop there. Don't forget to like the video and get notified. Click on the bell, get notified, and check out the other papers. Read this paper on the website and compare it to the interleukin 2 monoclonal antibody paper, which I think is much better. So I'll stop there and come back and join me next week. Thanks.